welcome to the fifth video module of the chapter Molecular Basis of Inheritance. In this video module, we shall be discussing about the translation and the operon, lac operon. Okay. So, what is translation? Translation is referred to the process of polymerization of amino acids to form a polypeptide. Right. There were genetic codes we discussed in the previous video. So, from the genetic codes, there is a production of amino acids, and from the amino acids, there are the production of different proteins. Right. So, what are the steps of translation? Okay, we shall be discussing about the steps of translation. Okay. So basically, translation is a multi-step process, right? The first step of the process is activation of amino acids. Okay. The first amino acid is activated. tRNA is activated. Sorry, my mistake. tRNA is activated. Then transport. Then production of amino acids. Okay. How it happens? Let's see. Let's say this is a mRNA, right? This is a mRNA. This is a 5' end. This is a 3' end. Okay. There are some start codons here, right? Like this. And over this, it is a 30S ribosome. And below this is a 50S ribosome. Okay. And here comes your tRNA, right? The codon arm of the tRNA. Okay. So here the complementary base pairs are there. Okay. So this is like U, A, C, right? So from this codon, there is a production of methionine, right? There is a production of methionine. Okay. Then it enters from here, right? Then there is outside another tRNA, which codon is like complemented to the next base pair. Let's say G, A, C, like this. Okay. So it will go from outside and it will come inside. So this is the entry point, right? This is the exit point. This is the site of acetylation. Basically, there are three sites, okay? And the ribosome shifts to the right side, okay? It shifts to the right side. And like this, there are different amino acids, right? Amino acid 1, amino acid 2, amino acid 3, amino acid 4, and these are called the peptide bonds. These are called your peptide bonds. Okay. So by this process, the new protein molecule is produced. Okay. You can go through the NCERT. I have marked this for you. Okay, no problem. Next is the regulation of gene expression. That is our lac operon. Okay. Okay. The process of translation involves what? Translation means RNA to protein. Option B. So basically, this is your video module over lac operon. Okay. I can guarantee you, no one can make you understand lac operon in this much short time. Okay. I will take very short time. Okay. Let's do it. What is a operon, right? Operon is a multi-gene system, right? There are multiple genes here, like this. So these are called operons. So in Escherichia coli, there is also a type of let's say operon, that is called your lac operon, right? So there is an operator here, there is a promoter here, there is an inhibitor here, right? And there are three structural genes Z, Y, A. Okay. So basically, inducer produces a repressor protein. A repressor protein. Okay. That binds upon the operator, right? Like this. And inhibits the lac operon. Okay. It is in the presence of. This is in the presence of lactose. Okay. This is the presence of lactose or abundance of lactose. Okay. Now what we do the when lactose is not present. Okay. Lactose is not present. Okay. तो जब lactose present नहीं रहता तब क्या होगा? It is the operon, right? This is the operator. This is the promoter. This is the inhibitor. This is Z, Y, A. Okay. So it produces a repressor protein, right? But when lactose is there, some bit of lactose, it binds to the repressor protein. It binds to the repressor protein and inhibits the repressor protein. Okay. That means lactose itself inhibits the repressor protein. Okay. So what happens? Key, the transcription starts from the operator. Okay. And then Z gene, Y gene and A gene gets transcripted and some enzymes are produced. So what does Z produces? Z produces beta galactosidase, which basically hydrolyzes the sucrose into glucose and fructose. Okay. Y means permease. Permease increases the permeability of lactose into the cells. Okay. What is A? A codes for trans-acetylase. A codes for trans-acetylase. Okay. So this is basically your lac-operon. Okay. So what they have said, ki lactose is the substrate for the enzyme beta galactosidase and it regulates the switching and off of operon. Hence it is termed as inducer. So lactose is called as inducer. This has been asked many times in your exams. Okay. This has been asked many times in your exams. Okay. So you can see the diagram here. In the absence of inducer, there will be repressor protein bound to here, so there will be no transcription. But when there is presence of inducer, that means lactose, right? There is lactose, so the repressor protein gets inhibited, that gets inactive, and the production of beta galactosidase, permease, and trans-acetylase happens. Okay. So see the question in lac-operon, the repressor protein binds to the operator. Okay, operator. So yeah, this was all about your translation and the lac-operon. Hope this video is not like kind of give a crystal clear concept, because it's a rapid revision series and you have to cover your syllabus as early as possible. Okay, so stay tuned with us. We shall be covering up all the topics in some few days, and do like and subscribe, and thank you, and visit our website for more details.